Excel students, or evening since you're probably watching this the night before lab. Welcome to the lab. Here we have our water heater, we have our radiation chamber, aka a microwave, centrifuge, microscope, and the plate reader. We wanted to stress how much the plate reader costs, so don't break it. So here we have the hood. This is the incubator where we keep all of our cells. So today we're going to be splitting cells. So I always want to be wearing gloves. And I always spray. No such thing as overspraying.
And that's a 70% alcohol solution.
Okay. So I will be splitting these A204 cells. So first thing we always want to do before and after we mess with cells is looking at them under the microscope. So this microscope is hooked up to the computer. And so here we have our A204 cells. So normal cell maintenance, you decide how much of the cells you want to take and put into a new flask. And so this one we're going to take 1/8 of the cells. So we're going to do a 1:8 split, and I'll explain that later. So we're going to turn on the hood. So these three switches, flip them all up. And very generously spray the inside. Again, no such thing as overspraying. Turn on the pipette aid. We're going to loosen the lid of the PBS and take off the lid for the pass-through pipettes. So everything gets sprayed before going into the hood except for things that have cells in it. Because there's a little vent up there, and if I spray it with alcohol, the alcohol is going to get in. Okay, so I need two things, my trypsin and the media for the cells, which you have to heat up in the water bath because we want it all to be warm to not shock the cells. So, take it out of the bath.
And each cell line has different media. You can see they're labeled on the front, and make sure you have the correct cell line.
So again, everything that doesn't have cells in it gets sprayed down before going in. So always spraying your hands if you touch anything outside of the hood. So our flask is in the hood. So when it lays in the incubator, it lays like this. So all the cells right now are adhered to the bottom or the back of the flask. So I want it facing me. First thing I'm going to do is I'm going to vacuum off the medium. So I loosen it, and I'm going to turn on the vacuum. And so it's very crucial when I'm handling the pipettes or anything else that's going to go into media or cells or anything that I only touch the very top of it to not contaminate the tip. So hold it like this, grab the end of the vacuum tube. Notice I'm only touching the tip of it. So from here, I can take off the lid, hold it like that, so I'm never setting the lid down to make contact with anything inside. And I want to put the tip here in the front corner because, as you remember, the cells are on the back side, so I always just want to touch the front side. I just tip it up, let it all run out. Okay, so from here, first thing I want to add is PBS. And PBS, all it does is just wash the serum off the front of, off the top of the cells. So, loosen that, and for a big flask, I'm gonna add 10 mils of PBS. So, whenever I open these, I never want to open it out here. I always want to have it deep under the hood. And when I open it, I want to open it where the side that I insert is what I'm handling. And if I touch the end or anything down here, I just throw it away and start over because I don't want any contamination getting in. So here, just going to insert it. Sometimes you feel a little click, sometimes you don't. And I pull it off, never touching the end. So from here, drop 10. So up, the top button is to go up in the pipette, the bottom button is to go down in the pipette. So up is up, get it up to 10. And I'm going to run the PBS down the front because I don't want to wash off any of the cells yet. From here I'm going to tighten up the lid. I'm just gently going to rinse back and forth, rinsing the cells off. A couple rocks back and forth, ready to vacuum off the PBS. So next thing I'm going to add is trypsin. So again, I'll loosen up my lids. Here I'm going to use a 5 ml pipette. Here you have 5s, 10s, and 125. So again, I open it with the side that goes into the pipette aid, never by the tip. So what also makes it easy is here you have like the 0, 1, 2, 3, 4 counting down. Makes it simpler if I have the side that counts up facing me. So a lot of times I'll turn it to where it's facing me, and that just makes measuring a lot easier. So I'm going to pull up 3 of trypsin. And notice, if I drew too much, once I pull it out of the bottle, I'm never going to put it back in. Just dump it out in a beaker or something. But once this leaves, I can't dip back in. And if I have a pipette that I dipped in something else, I can never go into a different container. So the trypsin, now what the trypsin's going to do is knock off the cells and basically just let them loose from the back of the flask. And so now I can run that down the back. Tighten up my lid and put it in the incubator for a few minutes. So our cells have been sitting in the incubator under trypsin now for about 5 minutes. So, what the trypsin does, remember at the beginning we said that the cells are all adhered to the bottom or the back of the flask. What the trypsin does is it breaks them loose. So, I'm going to look at them under the microscope to make sure that they did in fact all come loose. So, you see here that a lot of them are still stuck. Whenever I move it, some are floating, some aren't. So what you can do is just hold it firmly. Notice I'm never tipping it toward the lid, but I'm holding it firmly, and I'm just going to smack the back of it lightly. Notice it's never splashing out into the lid. I'm keeping it relatively still. After a little bit of that, now they're all floating.
Just make a note of how clumpy your cells are too.
Okay. So now that I see that they're all not adhered to the back anymore, now I can split them. So, splitting, I'm going from that flask to a new flask. So the flasks are in here. These are the big flasks. Always make sure to reseal it. So anything that doesn't have cells in it at the moment gets sprayed if it's going in to the hood. So that's our new flask. So we said at the beginning of the video that we are going to split this flask 1 to 8. And 1 to 8 just means we're taking 1/8 of the total amount of cells that's currently in this flask. So there's 3 mils of trypsin in there, and so if I add 5 mils of our media, then I'm gonna have 8 mils total. So now that I have 8 mils total, I can move 1 mil, which is 1/8 of the total volume, into the new flask, and so that's how I get 1/8 of all the cells from that flask into our new flask. So currently 3 mils of trypsin in there. We're gonna add 5 of our media to bring it up to a total of 8 mils in the flask. Again, I open up the pipette deep under the hood, not out here. I never touch the tip of it. If I touch down here, even if it's just a graze, throw it away, start over, we have plenty of pipettes. So making sure my lids are all loose. Notice this is the A204 media, which matches what cells I'm actually using. Bring it up to 5. So I add 5. So now I have a total of 8 in the flask. So notice Jesse said earlier to note how clumpy the cells were under the microscope. So these were fairly clumpy, and so all I'm doing is I'm just going up and down. And that's breaking apart those clumps, giving me individual cells not adhered together. One thing to really avoid is bringing it too far up into the cotton and into the pipette aid. Really don't want that, because Jesse's going to have to come in here and take it apart. Alright, so I've been mixing this for a couple minutes now. There's no such thing as too much mixing. Jesse wanted me to say something about bubble prevention. So once it's empty, I never want to keep on blowing out, because that's going to make all of it really bubbly, and it's going to make it hard to work with. Again, I never draw so far in that I'm getting up to here with it. So, should all be mixed now. No clumps. So, as I said before, there's 8 mils total in here, and we're going to take 1/8 of that, 1:8 split. And so these flasks, the big flasks, hold 30 mils. And so if I'm taking 1 of that into here, I'm going ahead and just putting 29 of the media, so that when I put in the cells, it all totals up to 30. So for this, I'm going to use a 25 mil pipette. Make sure my lids are loose.
And the 25 mil pipettes can actually go up to about 30, 35. You can see the negative reading.
So here I'm at 29. Into the new flask. So I have 29 in there, and I'm just going to take 1 from here into there, and we'll be done. So 5 mil pipette. Just 1 mil of the cells. So I have 1/8 of the cells from the original flask.
And you want to try and put the tip into the media so you're not dropping them too far.
There we go. And so, as I said before, anytime I work with cells, immediately before and immediately after, I want to see how they look under the microscope. So here they look nice and healthy. If I saw really big clumps, I would go in and just mix it again like we did earlier. And these should be good, so I'll just label my flask, put my name, the date, the cell line, and what we split it. So I would write 1 to 8.
And the passage number.
And the passage number. Passage number is just how many times it's been split or worked with. So since that originally said passage 10, let's write passage 11 on it and put it back in the incubator. And it should be done.